So let's move on to part 5, the last part of homework 6, which focus on cancer immunology and immunotherapy. So first, some background knowledge. Um, tumors, tumors do not exist alone in your body. It actually exists in an environment that is infiltrated by many types of immune cells, mainly lymphocytes or myeloid cells, and we call this environment tumor microenvironment. And the tumor infiltrating immune cells will try to eliminate the tumor cells. Uh, for example, the tumors on the surface of tumor cells, you will have, let's say, a complex that will present antigen to, say, an immune cell type like T cells. And T cells, upon recognition of this antigen, will try to eliminate the tumor cells. But we usually find that the tumor cells are not just going to sit there for being killed. It will suppress the function of immune cells through a mechanism called immune checkpoints. For example, it will on the tumor cell, it will have, say, PD-L1 ligand, which can bind to PD-1 molecule on the surface of T cells. And upon recognition, it will suppress the function of T cells. So to knowing this mechanism, many drugs called immune checkpoint inhibitors have been developed to prevent tumor suppression. So we can give them a man-made ligand for PD-1 or for PD-L1. So you will occupy the binding positions so that PD-L1 and PD-1 be cannot recognize each other since their binding site is already occupied by other ligands. And in this way, the function of T cells can be recovered and reactivated to kill the tumor cells. So for this part, I think you don't need any coding, but you need to do some exploration using timer uh, to answer a question that why the immune checkpoint inhibitor therapy doesn't work well in glioblastoma. Well, it works quite well in, I think, SKCN. Uh, I forgot what cancer type is maybe a skin cancer. And for question 1, you will be asked to compare the infiltration level of CD8 T cells, which are the effector T cells that kills the tumor cell. And you need to select the gene differential expression gene D tab under the exploration tab. And you just input CD8A and CD8B. They are gene markers for CD8 T cells. And you compare the infiltration level in glioblastoma versus SKCN. And in question 2, you will be asked to check the association between immune checkpoint expression and the CD8 T infiltration level in glioblastoma and SKCN. And you need to use gene under the immune tab. And just include the CD8 T cells as immune infiltrates. And a note here is that for PD-1, you should really include PD-CD1 because timer uses different name for them. And for PD-1L, it's CD274. And for question 3, you will be asked like whether CD8 T cell infiltration affect survival in glioblastoma versus SKCN. And for this question, just use outcome under the immune tab. And last question, I think it's just for question it's a question just for graduates, is that you need to combine your observations and findings from question 1 to question 3 and explain in a short paragraph that why checkpoint inhibitors do not work well in glioblastoma. I think when you get a result from Q1 to Q3, it's very obvious. Yeah, that's the end of the lab.